The project Three-spine stickleback is a key species of the White Sea ecosystem, history, structure, and main results. There is not only history, but also prehistory. And this is a citation from the book, which is called Animal Life, Жизнь животных in Russian. And it was a very known and very popular book in Soviet Union, published in this special volume on fish was published in 1971. And it was a phrase, it was a description of the stickleback in the White Sea, three-spine stickleback, the following description. In the White Sea, three-spine stickleback is a true marine pelagic fish. In Kantalaksha Bay, soon after the ice melting, stickleback massively approach shoreline. During the mass spawning migration of stickleback, water is literally getting dark of huge schools of fish concentrating inshore. And this description was done in 1971. And I first appeared in the White Sea in 1980. And I expected to see this picture because I appeared, first time came to the White Sea in the spawning season of the stickleback in June. And I tried to find this schools which is make the water dark of the backs. But I didn't find any stickleback. And it was a surprise. And when I first appeared in the Sredny Island, where the biological station of our university is situated, it looks like that. It's a picture from 1980, and there were no stickleback as well. So it was a surprise, and for some time we I forget about not completely forget, but so I forget about stickleback as a potential object for study. But in late 90s, the stickleback became appear more and more in the White Sea, and due to this, we started our project in 2005. The first sample was done in 2005. You see Mikhail and Tatiana Ivanova. So they we sampled stickleback with beach seine. It was a quite big and not convenient beach seine for stickleback. And year after, we prepared special smaller beach seine for stickleback, only 7 meters length, and it was much more convenient. Here you see historical picture how it is prepared. We need to choose location for study, and the first and very obvious location was a Sildenaya Inlet. You see how dense the seagrass there, and seagrass is a very important substrate, very important habitat for spawning stickleback, and it was the first our sample site. Then we decided to use also other habitats, because maybe one type of habitat is not very representative, and we decided to make our samples also in the shallow muddy lagoon, small lagoon. It's you see here the area of this lagoon. It's much less than 1 square kilometer. And we started sampling this lagoon in 2009. So very soon we were developing our methodology, and one of the important component of this methodology was photographing the stickleback. So from each sample we take about a half a liter of stickleback. It's about 100 fish. You make the photographs.
Dmitry Ludogovich,
Yeah.
sorry. Can you take your microphone near the mouth?
Okay,
Yeah.
this is better.
Yeah, thank you.
Thank you, Anna. Okay. So, and this is an example of first such picture which we use, and now we have almost 1,000 such pictures. So it's a huge information source for the study. And this is example of more modern picture. And this is we photographed not only adults but also juveniles. And quite soon after the starting of the project, we start historical studies, and because it was mostly because we remember that in book from 1971 it was such a expressive description that it was a period of very high abundance of stickleback. And after some research, historical research, we found that this description was attributed, in fact, to the 1930s. So it was a period when, as we knew later, it was a period of very high abundance of stickleback in the White Sea. But the author who wrote the book from 1971, he didn't even expect that that fluctuation of abundance of this species can be such high, and he just wrote this in very general terms. But the abundance of stickleback changed a lot in the White Sea, and you see here how it can be. It's the first picture from our study, historical study. But I will talk about this in much more details later this talk. In 2008, 2009, we did quite a lot of analysis of spatial distribution of stickleback in our area in Kyrgyz archipelago near the station. And you hear the abundance of adults. Here you see abundance of juveniles. It's quite a big difference because adults, they are distributed much more evenly than juveniles. Juveniles, you see that very distributed are very unevenly. Also, we did experimental study. Here, Elena Rybkina does experiment on preference of juvenile stickleback with different substrate, and we found that they actually prefer the seagrass when they a bit grown up. Then we in 2009, 2008, 2010 we got first money for research, for first project money, external money, and were able to organize expedition around the White Sea. So our project became the White Sea scale, and here you see the samples which we where we took stickleback and catch with them with beach seine. You will see results of these studies later. And yeah, actually here the very overview. And here you see the circles which reflect the abundance of stickleback, and it is very interesting that the highest abundance exactly in a place of our study, the highest abundance in the White Sea. But it is very abundant in other part of the White Sea also, as you see, except the eastern part of the sea. Then we also started to study ecosystem effect of stickleback and the trophic links and parasitology and so on. You will also know from today from other people, from my colleagues. We developed a tagging method. So we put the small plastic tubes to the spines of stickleback, to the back spines of stickleback, and it allowed us to get very interesting results. The first winter expedition took place in 2013, and the results of this expedition were quite unexpected for us, because this is the most common, the most abundant fish in the White Sea, which in summer it comprises about 95% of the biomass of fish in coastal area. In winter time, so we didn't find we didn't find only very few stickleback. So and that's one of them we only find 3. and in the lagoon. So in winter time, it's wintering places of stickleback are different. So places from we can't approach them until now. Also, we collaborated quite intensively with biochemical researchers from Karelian Research Center in Petrozavodsk, and this collaboration started in 2014. The first stickleback day was carried out also in 2014, and so much less people participated in this meeting that now, which I'm very glad that it's so. In 2014, again, we started project on visual asymmetries in vertebrates, and this project was led by Igor Malashchev, who unfortunately died about 2 years ago, and we also got quite interesting and important result for stickleback. In 2015, so we paid much attention, and until now we pay much attention to the age determination. Age is very important for our project and for project like ours, because it's ecosystem role of stickleback and the population dynamics of stickleback. But the analysis of age was quite tricky, and not from the first time we were able to read the age correctly, and it took some time, but now we can. 2017 we started our website, and this is one of the first pictures from that, and yeah, now it is much more developed and much more advanced. This year we spent a lot of time, last year actually, a lot of time to that. Our logo, it may look quite unusual for people outside, but we spent quite a lot of time discussing this in 2017, and this logo, this is a kind of citation of old petroglyphs in the White Sea area. So this is picture from petroglyphs. You see here, it's pictures on stones from about 6,000 years ago, and from our view, this kind of presentation of our logo says that we understand perfectly that we are all in the same boat. About 3 years ago, we got again external funding, grants from Russian Foundation for Basic Research and Russian Science Foundation, and we were able to improve our technical things. For instance, we bought motors, and it is a very important step towards for our project. 2019, we start to organize more experimental design in our biological station. It is quite primitive so far, but I think that it has quite a good potential for development. Unfortunately, last year we were not able because of pandemics, but now we plan to develop this further. 2019, we started our research in the Baltic Sea, so not only White Sea, but still most of research we do in the White Sea, but also there is some research in the Baltic Sea as well. 2020, due to COVID, there were no students in the White Sea, and only part of our team were able to participate in field studies, Mikhail and Tatiana Ivanova, and they did mostly studies in the open sea. So far we did studies only in coastal zone, but now we extend our area of our study, and it already brought some important results for the project. The structure of project in one picture. So this is two different parts so ecosystem aspect and population aspect. And ecosystem aspect include feeding of stickleback and feeding on stickleback, and there is overlap of this is a cannibalism when stickleback feed on themselves, and it's quite interesting and very important aspect in biology of stickleback. And Mikhail Ivanov will talk about this a bit later. Abundance of spatial distribution of predators and competitors. Yes, we studied them as well, not only stickleback, but we try to address most of species which are linked to stickleback, trophically linked to stickleback. Habitat preference of this fish, and also we start some studies on microbiome and parasites. And population aspects includes biochemistry, phylogeography, homing and site fidelity studies, behavior, response to different stimuli, behavior, migration on spawning grounds, embryology, morphological variation. And I and my colleague will review this later today. If to have a look on the available information of stickleback, so which may be expressed in number of publications as I did here, you see that the first information on the white stickleback is available since late 19th century, and of course it's very sparse information only about abundance, and most of studies, early studies was about abundance, but it was a period of in the 50s, 60s, when still it was quite a lot of stickleback in the White Sea. It was a period when researchers paid more attention to this fish, and some studies, but most of studies were done after 2000, and not only by our team, but mostly by our team. And in terms of space scale, it is of course it's mostly we did research in our area near the around the White Sea station of the university, but also we did research in the White Sea. And this is if to see this distribution range of the White Sea, you can see where our study area is situated. It's not far from the border of distribution range of the of the stickleback, fish-spine stickleback. And now some words about population aspects, which we saw in more details. So this is a life cycle of the three-spine stickleback in the White Sea, and the green is juveniles, and the brown is adults, and it's age 1, 0, 1, 2, 3. And the green shows the inshore, when they are inshore. Most of stickleback mature at 2 years old, but some of them mature at 1 year old, about quarter of the total amount. And you see that with time, of course, so there is some mortality. It's about 70% per year, and they live up to 4 or 5 years. We didn't see stickleback older than 5 years. Inshore, they spend only a small fraction of their life cycle, but this is very important for population and ecosystem point of view. Of course, this is spawning, and the first weeks of juveniles are very important for the further fate. Long-term changes of the population abundance of stickleback, it required quite a detailed historical studies, and it required quite a lot of field studies, because we already studied stickleback. We have a time series for 13 years. It allows some statistical analysis, and also we try to calibrate historical studies with our field studies. We try to simulate historical catches, when people mostly try to catch as more stickleback as possible, and because of that, it's not so easy to interpret historical study. But when you calibrate them with modern study, it will make it makes much more easy. And this is the last our sampling simulation of historical sampling in June 2019, and we got about 150,000 of stickleback in one hole of beach seine. This is very similar to what people got historically in 1930. And based on all these studies, we were able to create a time series of C-spine stickleback in the White Sea, time series of the abundance. You can see it here. You can see that the fluctuation in abundance are very high and approach about 2, maybe even 3 orders in terms of numbers, in terms of abundance on spawning ground. And the pattern are like that. So they were abundant in first half of 19th century, of 20th century. Then the abundance decreased. Then it was a period of very low abundance, less than 0.5 individuals per square meter. And then since late 90s, the abundance again increased. And this abundance quite well correlated with temperature. And it seems to be that temperature is the most important factor for stickleback dynamics. And here we try to correlate the abundance of stickleback in some particular year with temperature, with different lag of temperature back. So, for instance, when we correlate abundance of stickleback in current moment with in June, because we work in June on spawning grounds, with April of this year. So we see that there is significant correlation. The higher temperature in April, the more fish we have in June, and the same is with March. But this is, and it means that in this particular year, temperature right before the spawning ground, the spawning is important for abundance of fish. But we also found more distant in time correlation. So we found quite high correlation of the first of abundance of fish when they were 2 years old, with the abundance with temperature in the first year, especially temperature during the wintering. For instance, first wintering of stickleback has very high correlation with abundance of stickleback 2 years later. And it means that the temperature in winter is very important, very crucial for survival of juvenile stickleback and for the abundance when they are old. And also high correlation with June, with first months when they appear in embryonic period. But you have seen here only very weak correlation with winter time temperature, but it is just before under the ice. The water temperature doesn't change a lot, and so the correlation with winter period is very clear from the correlation in October and March, because this month they characterize the beginning and the end of the ice period. So this is this says that first wintering is a crucial for survival and abundance of stickleback in the White Sea. Some information about spatial distribution you will see in the next presentation by Tatiana Ivanova. I don't talk about this here. Some words about sex distribution, sex ratio of stickleback. Sex ratio is different from 1 to 1 quite a lot in the spawning grounds, and you see that we have 60% of males and 40% of 60% of females and 40% of males. And we studied we analyzed sex ratio not only on adults but also in juveniles using genetical markers. and found that in embryos, the sex ratio is about, doesn't differ significantly from 1 to 1. But in all the ages, there is a significant difference from the 1 to 1, and in some cases significant, and in some cases not significant. And prevalence of females, so females are more, and this prevalence we observe from the very early ages of stickleback, only when they are only a few weeks old. And the mortality of, and partly this imbalanced sex ratio can be because of females live longer, and female mortalities is lower in spawning grounds, and you see here that females. That mortality of female is during the spawning period is only 0.3%, but mortality of male is significantly higher, and this is 0.4%. It is not much, but probably it's indicative but it is the mortality which we can observe, and but in fact mortality is higher, and probably it is because we can't observe it. It happens in the open sea. And also you see here that mortality, predator-related mortality in stickleback is higher for males. In the sea, we have 35% of males and 65% of females, but in stomach of sculpin and cod, the main predators of the stickleback, we see the prevalence of males. So there is selectivity of these predators towards males. Analysis of homing with marking show us quite interesting results. The stickleback are able quite effectively found the home sites after they were taken from these home sites and marked and released in the center of the lagoon. You see that how in this four different home sites and with different experiment, you see that how effectively they can find the home site after about 1 day. Quite a lot of experiments were done on behavior of stickleback in the project on asymmetry. So we put males and females in one compartment of the experimental study, and analyze how they respond, how they react to the stimuli. Stimuli could be very different. It can be one male, one female, three females, and the different stimuli. And we analyze the response and got quite a lot of results, but I just show you one result. So how males and females respond to stimuli so here is stimuli, and here we analyze position and orientation of fish. And you see that males in this position, just in the front of this experimental experimental vessel, so they are much more, they orient towards the stimuli much more strideful than females. Females they just usually pass the stimuli. Very recent results on morphology. So I have to finish, but a couple of minutes more. This are new results not only for external people, but also for members of our team. So we analyze males of stickleback males from different spawning ground, and found that in the top of Kandalaksha Bay, the fish have not so high depth of the body, so the body is more fusiform than in males this more outer, more external part of Kandalaksha Bay, which are probably close to wintering, more close to wintering location. We don't know exactly wintering location are, but probably they are not far from this areas. So this pattern we found not only in scale of Kandalaksha Bay, you can see here scale 20 kilometers, but also in scale of Chupa Inlet, which is a part of Kandalaksha Bay. You see here scale 1 kilometer. We found that in the entrance of Chupa Bay, fish were more with higher body depths, and in the deeper in Chupa Inlet, fish were more fusiform. And this is and it is known that more fusiform fish, they are more able for longer migrations. And these differences in the shape in shape of the fish may reflect the ability to migrate. But the very interesting is that we also found differences between in this area between fish which are very, very close to each other. And this is next slide. You see here scale only 50 meters. This is lagoon where we spent we did quite a lot of studies, and this is place outside the lagoon, so it's distance maybe 200 meters. And you see that still quite big difference in body depth here, 100 here, 96%. And we interpret it as lagoon and sea are joined by the stream, by the small stream, and the fish who enter the stream and thus they can appear in the lagoon, they are more fusiform, and only more fusiform fish males they enter the stream because more fish with higher depths, body depths, they don't enter the stream. Also, I just show you some studies on biochemistry, especially lipids, and we found that there is a heterogeneity in amount of omega-3 lipids in omega-3 fatty acids in stickleback in our area, and this can be related to different feeding spectrum of juveniles in different size of Kandalaksha Bay, in Sidinai Inlet and Klishkovy Lagoon. So in the inlet they have more Helicostomela subulata. It's infusoria which contain more fatty acid, and this may be kind of heterogeneity which may be related to survivability of fish in the winter period. Quite a lot of other biochemical results, and also we get quite a lot of genetic. We did quite a lot of genetic studies, but so far not much of it published, and we are now working hard on publishing this data. And ecosystem aspect, you will. I just refer to my colleagues. So yeah, important to say that stickleback is very abundant fish, as the white sea I already told, but it is also fish which forms worst waste of the ecosystem, which means that together with herring, they control most of energy between plankton and top predator. Top predator are cod and sculpin, navaga, and some birds. And Anna and Anastasia talk about the stickleback and trophic chains, and Mikhail Ivanov will talk about role of stickleback and horizontal transfer of substance. And just one last slide about parasitology. So we did quite a lot of parasitological work and got quite a lot of results, mostly on juveniles. And acknowledgements to our sponsor, Russian Science Foundation, Russian Foundation for Basic Research. And this is, thank you for your attention.